All right, good morning, everyone. Thanks for joining us today. My name is Rachel Lanis, and I'm a technical applications scientist here at Cellular Dynamics International. I work primarily with the iCell Neurons product, both in application development as well as providing technical support both internally and in the field. So today, this morning, we're going to be talking about the iCell Neuron product and its use in bringing relevant neuronal biology to the bench. So before we get started, though, I'd like to just go over a couple of the logistics of how to submit questions for our Q&A session, which will follow the presentation. So on this next slide is just going to highlight the areas that you need to go to in order to answer a submitted question. Oh, so here we are. On the top of your computer, you should be viewing this little bar up here that says View my desktop, and once you come here, it'll open up this little menu at the top of your screen. Then what you'll need to do next is go over to the right, and you'll have an icon for submitting Q&A. So you can bring that up and click on that icon. Once you've done that, you'll have a little box open up, and you'll have a field in which to type your questions. So feel free to submit questions throughout the presentation, but just keep in mind that we will answer all of these at the end of the webinar. All right, so now we'll get started with the presentation. And just with a brief overview of the outline, we're going to cover the CDI technology, who we are and what we do here at CDI. Then we'll move into the iCell Neurons Characterization on how we've developed our product. Then we'll go into the iCell Neurons applications, cover some of the things that we've done internally with our cells and how we're expanding that portfolio of applications. And then finally, we'll touch on some publications that have come out within the last year on the iCell Neurons product. Now to begin, Cellular Dynamics International is the world's largest producer of human iPS cells and iPS cell-derived cell types. We're headquartered here in Madison, Wisconsin, with a total of about 105 employees, about of which 80 scientists, excuse me. So you can see we're very heavy R&D staff and really put an emphasis, a strong emphasis on the research that we're doing in the development of new products. With those 80 scientists, we actually have a total of approximately 400 years of experience in the human stem cell field, which is quite a lot in a field that's only about 14 years old. On top of that, we have greater than 460 patents, which are either owned or licensed, and therefore allow all of our customers the freedom to operate and really pursue the research that is important to them without being hindered by any legal matters. As a company, we really have 4 core competencies that we focus on in the development of all of our products. These are the creation and culture of the human iPS cells. Now this is, it's really not a trivial matter actually to develop and culture new human iPS cell lines. The maintenance really requires a significant amount of time in order to do this and to really maintain them well. We are able to create and culture these cells from really any genetic background, whether it be a normal or a diseased phenotype. So it expands and opens up the diversity of human biology. Secondly, we're able to then genetic engineer any of these iPS cell lines. And this includes incorporating lineage and pathway-specific markers. This can be done in the iPS cell line to allow for the differentiation process to be done, or for specific assays after the differentiation process and in the endpoint to assess functionality. Also, we're focusing on the development of new differentiation protocols. We are currently able to differentiate cells from all 3 germ layers, and while our product portfolio at this time consists of just a handful of products, we're constantly not only improving the differentiation process for these particular products, but we're expanding and differentiating new cell types. And finally, we are able to manufacture human iPS cell-derived cell types at a very large scale, at an industrial scale, in fact, so that we're able to scale the production of these cells for any high throughput screening process. And as a highlight, CDI was actually awarded the 2011 gold winner for the Wall Street Journal's Technology Innovations Award. So it really speaks to the competencies and the abilities of CDI as a company and what we're doing in the stem cell field. Now, as I mentioned, we have a handful of current products that are available. Our iCell Product Portfolio consists of the iCell cardiomyocytes, which were the first commercially launched product here at CDI in late 2009. This was followed in 2011 by the iCell endothelial cells, as well as the iCell neurons, which we're talking about today. And finally, we have iCell hepatocytes, which are actually in a pre-commercial release state and are shipped currently as a live cell product. We're continuing to research the cryopreservation of these cells, but to date we have shipped more than 20 billion cells since July of 2011. Now, on top of that product portfolio, we also have recently launched a custom service called MyCell. Now, what this involves is that we are able to take samples from any customer and reprogram those samples into iPS cells. Now, this reprogramming process that's been optimized for MyCell is an episomal or footprint-free method. It's been optimized for small amounts of peripheral blood, but we're also able to take other samples, for example, fibroblasts, and it's done in a feeder-free and defined condition. So really, we can, if a customer has a panel of, for example, 10 patients with a different disease phenotype or disease background, we can take samples from all 10 of those patients, reprogram them into iPS cells, and then from those, do any genetic engineering that that customer might require or desire in their cell types, and then finally do the terminal differentiation. Now, in the case of a customer interested in a panel of Alzheimer's patients, we'll then differentiate those into our catalog neuronal product. Now, in order to generate and really do the work that we do here at CDI of the iPS cell reprogramming and terminal differentiation, we really focus on three key characteristics for adoption of our technology into new organizations. And those are quality, purity, and quantity. Now, to begin with the quality of our cells, we really strive to make sure that all of our cells exhibit the key cellular characteristics and recapitulate normal human biology, so that the iCell neurons you receive will not only look like human neurons, but they'll also function and act as you would expect normal human neurons to function in culture, so that they can be utilized in endpoint assays. We manufacture our cells in a very reproducible manner, so you're getting lot-to-lot consistency and don't need to worry about optimizing each new lot of cells for an assay that you're interested in. And they also come from a known and relevant genotype. Now, to move on to the purity of our cells, we have optimized the differentiation process as well as the maintenance of our cultures over time to obtain very high levels of purity in our cultures. We require all of our cells to be greater than 90%, but oftentimes they're much more closer to 95 to 99% pure. And in the case of our iCell neurons, they're very, very typically around 90% pure. But the reason that this is important is that as we're differentiating our cells, we are eliminating the non-target proliferating cell types through this process, so that we can have a very high level of our pure final terminal cell type. And again, this purity is then maintained with the optimal culture conditions that we've created in our media and defined or are outlined in our protocols. And finally, we look at the quantity of our manufacturing process. And as I've said, we've brought this, made this a very industrial scale manufacturing process so that we're able to produce enough cells to support any high throughput drug screening or safety testing. And as an example, our current iCell cardiomyocyte manufacturing process, we yield 1 billion pure cardiomyocytes per day at full capacity. Now that we've gone over the basics of CDI and the iPS cell technology, we'll move into the characterization of the iCell neurons. So as we've kind of highlighted all of our different cell types, they come as, the iCell neurons will come as a cryopreserved and ready-to-use single cell suspension. So they're shipped on dry ice, and they'll come received to you to be received and stored in liquid nitrogen until you're ready to use them. So your workflows don't require you don't have to build your workflow around the availability of the cells. You can plan the use of the cells around your own workflow and your own time. Again, they're terminally differentiated, so they're not a cell line. They're not going to reproduce or grow and divide in culture. However, they can be maintained for long-term in culture with the proper maintenance and care. Again, the purity is going to be very high, oftentimes between 95 and 98% pure. The cells themselves are going to be platform agnostic and compatible with existing assays and instrumentation, and we're continuing and always exploring new assays and instruments to expand the use of our cells upon. The cells will come actually as a complete product solution, so this will include not only the cells, but also the culture medium to maintain the cells in and maintain that high purity, as well as the protocols for use, thawing and handling. And we also have on our website some application protocols for use in different assays and on different platforms. And just to reiterate, these cells are produced in quantities to meet any demand, so we're not held up by production or manufacturing. We have cells in the tank ready to ship out at any time. As we developed the iCell Neuron product, there were some key neuron characteristics that we knew we had to focus on in order to have a product that customers would want to use in their research. These include the morphology. So as we can see from the stained iPSC image on the top right here, we have nice bipolar and multipolar neuron outgrowth from all of our cultures. This image is from day 7, but we'll see on the next slide that even at day 1, within 24 hours, we have nice neuronal neuron outgrowth already developing. We've also assessed our cells with different molecular markers to look at general neuronal markers as well as some subtype-specific markers. This list here is certainly not exhaustive, but just highlights a few that we've utilized, including the beta-retrubulin and MAP2 as general markers, the VGAT and VGLUT and tyrosine hydroxylase for the more subtype-specific markers. And finally, we assess the functionality of our cells through some different endpoints looking at electrophysiology as well as neuron outgrowth and sprouting. And what we see from the images here at the bottom is that the iCell Neuron product is actually a mixed neuronal cell population. It consists primarily of the GABAergic and glutamatergic neurons at about a 50-50 ratio. And there is, in fact, actually very, very small amount of dopaminergic neurons in there as well, yet that is less than 1%. So we really do call this a mixed population. But that mixture there is very typical of the forebrain region. And again, to highlight the purity of our cells, we just show that the purity is assessed by beta-3-tubulin positive stain and nestin negative to show that there are, in fact, no progenitors in this population. And we see here that purity is very high at 98% post-thaw. And finally, just to highlight again the industrial scale of the manufacturing process, this 50 ml conical contains a 5 ml pellet of pure neurons, which is equivalent to about 4 billion neurons, or 4% of the human brain. And this is the result of just a single manufacturing run. So it really does speak to the capabilities of our process. And to further characterize our cells, we've looked at the post-thaw morphology over time as well as the corresponding purity of those cultures. You can see, as I already mentioned, that at day 1 post-thaw, within 24 hours, in fact, we're able to see very nice adherent cultures with good neuron outgrowth already in 24 hours. And you can, in fact, see this within just a few short hours after plating your cells. However, we recommend waiting until 24 hours to look at your culture. And you can see the corresponding purity for this culture is about 98%. Now, over time, as that culture is maintained from day 5 to day 19 and on, that neuronal network will continue to expand and become more extensive and more sophisticated with time. So much so that at time points of 2 to 3 months in culture, it's so dense that you really can't see the bottom of the plate. And we see that the purity is maintained above 90% as we take those cultures out longer. So we really do have a nice, highly pure population post-thaw that has a very efficient cell recovery within 24 hours. And that network will just continue to mature and develop over time. And to further then characterize the subtypes within our mixed population, we have some nice staining here just to show the presence of the VGAT and VGLUT. So we have the very nice punctate staining along the processes that shows we have the machinery and the functionality, or the machinery to be there to show the functionality that we'll see later on, and the presence of those two subtypes of the GABAergic and the glutamatergic neurons here. And we also see that we're able to stain with GABA to highlight those specific subtypes, along with the general stain of MAP2. And finally, in some of our functional characterization of the cells, we assess the electrophysiology of the cells by a single-cell patch clamp analysis. And what we're seeing here is that iCell neurons are able to generate, and we're able to record both evoked and spontaneous action potentials off of our cells. And we have a very nice mature-looking action potential here with the depolarization of both 0 millivolts and then back down to the resting membrane potential within a repolarization. And in the panels on the right, we're actually assessing the specific major ion channels in our cells, sodium, potassium, and calcium. And we're seeing a very nice dose-dependent blocking of each of these currents with the known and respective antagonists. Now, in the case of sodium, we're looking at the treatment with TPX and see a nice blocking of that current. With potassium, we treat with TEA and also see a nice dose-dependent decrease in that current, or blocking of that current. And with calcium, we have a single response to a nifedipine or calcium antagonist there. Now, once we've then functionally and characterized the subtypes of our cells, we're now using them, actively using them in numerous applications and platforms to further assess the functionality and utility of our cells. And this is something that we're constantly working on and expanding in time with our cells, both internally and through collaboration. So we'll highlight several of the endpoints here that we've used to assess the functionality. But these certainly, excuse us for 1 minute as we're having a little difficulty here. Okay, sorry about that, everyone. So as I was saying, this slide, we're just highlighting several of the endpoints that we use to assess the functionality of our cells. This certainly does not capture everything, but it does highlight a few that we've worked on in the last year. These include the electrophysiology via, again, the single-cell patch clamp, as well as the multi-electrode array platform from both multi-channel systems, as well as Axiom biosystems, to assess the overall network activity of our cultures. We've also looked at the cytotoxicity of our cells via some cell-based assays. We can assess the mitochondrial health of our cells, or the bioenergetics, on the Seahorse Biosciences platform. And we're able to detect levels, normal levels of proteins of our cells that can then be used for disease modeling. We'll get into this on a later slide. We've done some receptor signaling analysis on the Flipper Tetra system. And finally, we've assessed neurite outgrowth both on high-content imaging as well as through some real-time neurite outgrowth analysis on the SM Biosciences platform. So as I said, we'll get into a few of these in more detail in the next few slides, but it's certainly not an exhaustive list. Now, with the multi-electrode array platform from Axiom Biosystems, we're able to really, it's different from the single-cell patch clamp in that we're able to assess the activity of the network as a whole. So all of our neurons interacting together. And what Axion's platform really allows us to do is work in a relatively more high throughput manner than the single cell patch clamp. In this case, their Maestro system is a 48-well platform, and we're able to directly culture our cells on these plates and monitor them out to longer term in culture. But we do see by day 4 that we have spontaneous activity, indicating functional networks of our cells at this time point. And that activity then we're able to modulate with specific compounds. In this case here, we're looking at a variety of agonists and antagonists, 2 different ion channels, and their impact on the mean firing rate or the overall spontaneous activity of our cultures. And we see that, for example, with some known GABA-A receptor antagonists, we're seeing an increase in the activity that we would expect by inhibiting or blocking the inhibitory neurons. We're also able to treat then with some known blockers or agonists and antagonists of, for example, the sodium channel with lidocaine, and we see a shutdown of that activity, as well as with GABA and TEA. So all expected responses being detected, showing the utility of our cells on the NEA platform. And then finally, just to highlight not only the changes in the overall activity, we're also able to see changes in sort of the morphology of that activity, or really the spike rates and what those look like. So in our controls, we see sort of just random spiking here, but when we treat with gabazine, we're not only seeing an increase in that spiking activity, but also the change in that phenotype where it becomes more of a bursting pattern. So we really see utility and functionality of our cells on the MEA platform. We're also then able to assess toxicity by high content imaging. On the right, we're looking at just the overall integrity of the neuronal network and that neurite outgrowth. Now, as I previously said, we see very nice neurite outgrowth beginning within the first 24 hours of plating our cells. And that network can then be either increased, enhanced, or degraded with the treatment of different compounds. In this case, we're breaking down that network by treating with known toxic compounds. And we see the expected EC50s in both of our graphs here on the mean outgrowth per cell as well as the mean branches per cell. And then just as a visual, we have here a dose response to mitomycin C showing the degradation of the neuronal network as we increase the concentration. We're also able to assess the mitochondrial membrane potential via high content imaging. We see with a nice control culture, we see some very nice staining with JC10. And then after a very brief treatment with antimycin A, we see that mitochondrial membrane potential being degraded and depolarized. And finally, we have this nice, again, graph showing a dose response here to both malinomycin and antimycin A to the mitochondrial membrane potential, indicating oxidative stress to our cells. And we also realize that many people are going to be interested in using our cells for functional assays for discovery, including reporter assays. So we've put some focus and emphasis on assessing the transfectability and transducibility of our cells. At the top, we're looking at some transient transfection of reporter molecules using constitutively expressed TFP. And we've really developed a nice standard workflow here where we can simply thaw and plate our cells, transfect in that reporter gene, maintain the cultures for however a brief period of time you'd like, and then potentially induce that pathway and measure the activity. And with the image on the right, we're seeing some very nice expression of that GFP, and we're finally able to get about 25 to 30% transfection efficiency internally. Now, alternatively, then, we've looked at the transducibility of the target gene in our cells, and we find that they are able to be transduced here with an AAV RFP, and we see with the time course at an MOI of 60,000, we see an increase in that efficiency from 32% at day 10 up to 75% by day 25. So this really is showing the utility of our cells to be worked into a reporter assay, functional assays. And then in collaboration with Mesoscale Discovery, we assessed the levels of two different proteins, the tau and beta amyloid proteins, that are very closely related to Alzheimer's disease, showing us the utility or the potential to utilize our cells in disease modeling. And what we found here, start with the total tau expression and the phospho-tau expression as lysate assays. We're showing essentially the consistent expression level or detectability of these proteins that is consistent not only from one lot to another lot, but also from two different time points. So we assess our cultures at 7 days post-thaw and 14 days post-thaw and really see nice consistent levels of the tau protein in our cells, and this is maintained with the phospho-tau protein as well. And similarly, we see a nice consistent expression of the A-beta proteins and the beta amyloid proteins as well across multiple lots and different time points. And the only thing to point out that's different here is that we have an increase or doubling in our density of our cells, thereby showing a doubling of the proteins expressed here, as these are conditioned medium samples for the beta amyloid proteins. So overall, what we see is that iCell neurons are producing protein levels of tau and beta amyloid that are typical and expected of a normal adult. And these levels of expression are stable over time from 7 days to 14 days post-thaw, and they're reproducible between lots, showing the consistent performance of our cells and the consistent expression of proteins in our cells, and the potential utility then in disease modeling.